Okay, today we're going to do the pGLO Bacterial Transformation Lab. And so what we want to do, we want to put our lab coats on, our gloves on, and then we're going to clean off the countertop with some simple greens to start with. And then we're going to use rubbing alcohol to sterilize our countertop to minimize contamination, so aseptic technique. And what we have here is we have some transformation solution and some slurry broth. We're going to leave that sitting on the ice for now. And then in our trays over here with all of our supplies, we have 2 empty tubes. We're going to label one of those plus and one of those minus. So plus, I'm going to put a plus on here. I'm also going to put a plus on the top. I'm going to label this one negative or minus pGLO and negative sign on the top. The first step is to add 250 microliters of the transformation solution to each of our tubes here. So I'm going to take a sterile transfer pipette, and our little cheat sheet on here tells us that 250 microliters would be up to here. So I'm going to take transformation solution, the calcium chloride, and I'm going to add 250 microliters to the plus tube, and I'm going to add 250 microliters to the minus tube. And then these need to go on ice. The slurry broth doesn't need to stay on ice. So we're going to put those 2 tubes on ice. And we're going to discard our pipettes in the biohazard bag. And we want to then add our bacteria samples to each of our petri dishes in the calcium chloride. So we're going to take our starter bacteria, so E. coli bacteria. We're going to open up our inoculating loop bag, which is pre-sterilized for us. And we just really need 1 colony of bacteria. So we want to try and find just 1 little circle there on our petri dish. So got one of those and just a little gentle swipe. And then put that in the liquid, and we kind of want to stir it around to get the bacteria off of our inoculating loop. And then we need to get another colony for our negative pGLO. So I'm going to just scoop up bacteria there, colony, and we're going to swirl it around in our petri dish. I'm going to close up my starter plate, put that away. And then we're stirring to get the bacteria into our solution. I'm just spinning it like that. Go in the biohazard bag. I'm going to close my minus tube, and then I'm going to add the plasmid to just the plus tube. So I need another inoculating loop. My plasmid has been sitting here on ice. So I'm going to take the plasmid out, and I want to dip the loop into the liquid. And I want to make sure I can see the little bubble wand cover on that. And then I'm going to stick that into the plus sample. I want to make sure it goes right directly down into the liquid. And I want to spin it to mix up the plasmid with the bacteria that's in the liquid there. And I can close that. And these 2 need to sit on ice for 10 minutes. So I'll start timing, 10 minutes. while we are waiting the 10 minutes, it does tell you in the instructions to do a couple things with the UV light. So if we turn on the UV light and we shine it on the plasmid, we can see it's not the plasmid that's glowing. And if we also shine it onto our starter bacteria, the starter bacteria does not glow to start with. We want to try and transform it to see if it will start glowing after it takes in the plasmid. Okay, also what we want to do while we wait and that's sitting on ice for 10 minutes, we can label our petri dishes. And so these are color coded so that we don't have to remember exactly what is in all of them. And so we have two red ones that are color coded red. We've got one that's kind of yellowish and one that's kind of purplish. The two red ones are Luria broth with ampicillin added to it. The yellow one is just the Luria broth, and the purple one has the Luria broth, ampicillin antibiotic, and the arabinose sugar in it. So what we're going to do is the Luria broth and the LB amp plate, those are going to get labeled minus pGLO, and our LB amp and the LB amp aria, those are going to get labeled with the plus pGLO. So we want to label them on the bottom where the agar is growing, or the agar is, so that when the bacteria grow, the lids don't get switched. And so we know what's on each plate. So this is going to be minus pGLO, and this is going to be minus pGLO, and this is going to be plus pGLO, and then this is plus pGLO. And after we have transformed our bacteria, we're going to put the transformed bacteria onto the petri dishes. The transformed ones will be here, the non-transformed will be there, so we can compare those. Okay, that is almost 10 minutes. So what we need to do is we need to go over to our hot water bath. So we're going to take our samples to the hot water bath, and we need to heat shock for exactly 50 seconds. So it's going to go in the hot water bath at 42 degrees Celsius for 50 seconds. So we're going to start now. And so they're just going to float around in there for 50 seconds. And then it will go immediately back on ice. Okay, that's 50 seconds, so it goes right back on ice, and then we can put it down. It needs to sit back on ice for 2 minutes. And after 2 minutes, what we're going to be doing is we're going to be taking the Luria broth and we're going to add it to each of our tubes. So again, the one has been transformed, the other one has not. So I'm going to get my pipettes ready. And open like a cheese stick. And the Luria broth. Open that up. And so we're going to add again 250 microliters to each of these tubes. I'm going to start with the minus tube. It's been about 2 minutes. So again, 250 microliters of the LB. Let's put that in there. Plus one, get a new pipette. Again, 250 microliters. And that into the plus tube. And that in there. And then I believe it says to gently flick it to stir. So we want to make sure that it all mixes up in there. Gently flick this one. And then it can sit at room temperature for 10 minutes. I know that's only been 5 minutes, but we're going to go ahead with the next step. But you would normally wait 10 minutes to then transfer 100 microliters of each of these onto our plus and minus tubes. So these ones here are our minus Petri dishes, so we've already labeled those. And so we want to now take a new transfer pipette, and the minus, we're going to put 100 microliters onto each of these. So again, if you didn't flick it before, you want to flick it to mix it. And then we're going to just squirt 100 microliters, and so that's up to that first pump there. And then 100 microliters. Pump there, and so we're going to squirt that onto there. And close the lids real quick. And then we're going to do the same thing with our plus tube on these two. And again, 100 microliters into each. Okay, the next thing that we need to do is we need to spread that bacteria around so it can all get a surface area to grow on. So I'm going to take one of these and spread my minus ones around. You're just zigzagging, turning and zigzagging, turning and zigzagging to kind of spread it all over the place. And since they're both negative, I'm going to spread with the same inoculating loop, but you could use separate inoculating loops for each of those. And then I'm going to close those up, and I need another inoculating loop for these. And we'll start with the red one here. Spread that around, zigzag back and forth, zigzag back and forth, turn and zigzag some more. And then I'm going to do the same thing with the last one. Zigzag, zigzag to spread it around. Then what we want to do is we want to stack these up on top of each other, and we're going to tape them all together. And label it with our name, our date, and a period, so that we can find them again. And we're going to turn them upside down because we want the agar on the top. That way when humidity accumulates in the petri dish, any liquid would drop onto the lid at the bottom so that the bacteria don't get sloshed off and so that they can survive and grow. I'm just going to write the name on it and the date. And then we're going to put those in the incubator. So let's walk on over to the incubator. Take our samples to the incubator here. Up, and we're going to stick them on the shelf. And then we're going to close that up, and we'll come back in 24 hours to see what grows and what doesn't grow. Okay, our bacteria have been growing for 24 hours, so we're going to take a look and see what we have, what grew and what didn't grow on which of our petri dishes. I'm just going to take the tape off. So definitely look like we've got some growth on our plus plates. This one is our pGLO plus amp and our pGLO amp plus, and then that one's got the ampicillin and the arabinose. And then here's our minus with the antibiotics and the non-transformed. And here is our lawn of bacteria. And if we shine our UV light, there's a little one that comes with the kit. You can see it's glowing. I have a brighter one so we can see a little bit better. So definitely getting glowing bacteria. On this one down here, that one has just lauria broth and ampicillin, so the bacteria transformed and now have the ability to survive in ampicillin, but they're not glowing. I'll shine the flashlight on them. They don't glow because they don't have the arabinose sugar in it, and the arabinose sugar is the switch that causes it to glow green with the fluorescent light. Shine it like that, we can see even better the glowing. So no glowing and glowing. So really good results this year. And then what we could do is we could count our colonies and we could calculate our transformation efficiency. That was the word I was looking for. We could calculate our transformation efficiency once we've counted our colonies.